All right, so thanks everyone. I'm going to be talking about different part of the world, Africa, which is in some ways one extreme in terms of genetic diversity, but I'm going to be talking about lower transmission areas of Africa, which at least traditionally are considered to be areas of higher population structure and lower genetic diversity, although it's not necessarily the case, as I'll show you to some degree here. All right, so I'm not going to go into too much detail about the rationale for this. Everyone is here, so clearly people are interested in parasite movement. But I just wanted to make the point that at higher transmission areas, big questions about how much malaria there is, how it varies in space and time are really important. And that some of the more granular questions about parasite movement and whether infections are local or imported are really more important as transmission goes down. We really want to know what the focal hotspots are, what the high-risk populations are, and patterns of transmission are not so important in areas where the parasite prevalence is approximately 80 or 90%. Everyone's just infecting everyone else. But when transmission is relatively low, we have the opportunity to target our resources. All right, and so because measuring movement is more relevant, we want to define what specific questions about the movement are the most relevant, right? So one of them is, are cases coming from somewhere else? Are they being imported? And what you define as importation can really depend on, what your question of interest is. So you could find that as a village or a district or a country or perhaps even a region of the world. So your tools will depend in large part on your spatial scale. And then, to what degree are are parasites being brought in from somewhere else? Because everything else is local, and that's going to require you to do something in those areas that's potentially different than what you do for imported cases, right? And for local cases, you want to know over what spatial scales this transmission is occurring, and furthermore, are there specific patterns? For instance, discrete islands of malaria transmission that can be tackled one at a time, which makes things much more operationally tractable than trying to tackle an entire area at the same time. So I talk about nerdy stuff at home as I do at work, and I asked my daughter who's 10, to kind of draw a picture relevant to the things I've been talking about. And I think there's two salient features of this picture. One is that despite only being 10, she recognizes the critical importance of missing data and how that's really detrimental. And another thing, I really need to sit down and talk with her when I get home and have a father-daughter talk about the use of pie charts in statistical presentations. So that's a separate issue. All right, so at fine scales, I'm going to propose that one of the tools that might be useful is actually connecting the dots between individual infections. And this is acknowledging that we're not going to capture every case and we're not going to capture every linkage directly, but if we attempt to track that spread from person to person, there's a lot of information that we can gain. And what I've drawn here is a very stylized transmission network. We've got different infections, not different parasites, because these are people that might harbor more than one parasite. In fact, the majority in sub-Saharan Africa do. We have some missing cases here. And what we're trying to do is really track relationships between hosts, not between individual parasites per se. And ideally, we'd incorporate temporal, spatial, missing data, for instance, asymptomatic carriers, and allow for superinfection and polyclonal infections, which we know is a big deal. So my son did not want to be left out of the game and actually produce what is probably a much more realistic example of what a family tree of malaria parasites would look like than my simplified schematic over here. And in particular, he's got some severe malaria with clogged blood vessels and just kind of a range of diverse parasites, which is really, I think, well capturing what's going on in parasites populations in sub-Saharan Africa. All right, so this is just a real schematic to illustrate something I think was nicely discussed by Caroline in the last session, and probably incorporates a number of the different methods that are going to be talked about here. So, at larger scales, we don't necessarily want to know about individual linkages between parasites, but we want to look at connectivity between communities, right? And there's three important things that I tried to illustrate on this schematic here. So the first is that if you look, everyone's got more than one infection. And this is the rule rather than the exception, even in very low transmission areas in Africa that we've looked at. And the important thing about this is if you're limiting your analysis to the monoclonal infections, you might miss something or get a very biased result, especially in areas where importation is important, because the imported cases are much more likely to have higher multiplicity of infection than the local cases. All right, so now let's divide this into our two populations. And the second thing I wanted to point out is that if you use some standard population level metrics, such as FST, at least in this example, these communities are going to look identical. So again, in this example, the FST is 0. There's the same population frequency of all the different types of parasites, right? But if you instead look at differences between individuals within and between the populations, you can see striking differences. In this case, entirely driven by the multiplicity of infection. Without that, we wouldn't actually see any differences between this community. So you can actually potentially leverage the high MOIs to tell us something about the connectivity within and between groups. All right, so I'm going to quickly go over a couple of different studies that we've done and just reviewing the data and sample collection for all of them up front. So we did a pilot study in Zanzibar a few years back. We essentially worked closely with the malaria control program and 47 health facilities that are marked by little purple crosses on this map, and we enrolled about 2,000 cases over a period of a year spanning 2 malaria transmission seasons from 47 health facilities. We did a travel and residence questionnaire. It was a study question, but it was really well integrated with the health infrastructure. The second study setting is Namibia. This was something that was in addition to what was already going on at the health facilities. We did a very quick and dirty travel questionnaire, and the entire questionnaire is on this little barcoded sticker that we stuck on an RDT. The people in the health facility just ticked as quickly as they could the residents and a bit about travel history, stuck it on the RDT, and we just collected all the used RDTs from the positive cases. And for this study, we had a single study coordinator and a vehicle, and they went back and forth between 24 health facilities. So just to demonstrate there are ways you can utilize existing infrastructure to get very dense sampling if that's what you're trying to do in an area. And then the third study setting is Swaziland, where we're collecting dried blood spots from all active and passive cases. And this is something that's been integrated into the malaria control program. And we have some planned studies at different scales in Mozambique, Botswana, and South Africa. All right, so I'm not going to talk much about the genotyping methods, except to say that all the data I'm going to present now are based on very simple technology. We're using 26 microsatellites. And one of the main reasons that we've stuck with microsatellites is because we can genotype them at very low parasite densities, which is important for the asymptomatic infections. And they do really well when you have polyclonal infections, because you can detect which alleles are present. There are some other ways to do this. So we're evaluating some methods for doing amplicon sequencing, using molecular inversion probes, with collaborators Jeff Bailey and John Giuliano, and also Alison Kuchta, who's a postdoc of mine, has a poster on Wednesday where we're trying to use CRISPR-Cas9 to target amplicon sequencing for specific areas. And this essentially gives you the same type of data that you can get from microsatellites, where you have more than just 2 potential variants. So if you've got 4 or 5, 6 different parasites present, you can still potentially get some information, whereas with SNPs, that can potentially be limited if you only have a heterozygous call. We're also working on some ways to try to phase these data, even if we don't have very dense sampling across the genome. All right, so really important is what you're going to do with the data. And I think Alyssa mentioned this earlier, but the type of analysis that you're going to do really depends on the spatial scales and the population level diversity. So for the very focal transmission, when we're trying to really link these dots together of different infections, we have a model that's really consistent with the processes that shape genetic diversity at that scale. So not mutation, but really reshuffling of standing variation through sexual recombination. And it's based on a few key factors. One is importation from genetically diverse source populations. And if you have allele frequencies from these different source populations, you can potentially identify where they've come from. We assume that there's some sort of bottleneck between transmission, and some of that variation is lost in the absence of superinfection. And then thirdly, that there can be superinfection, which then enables reassortment and recombination. And this is work that's really been spearheaded by Alex Perkins at the University of Notre Dame. Some things about this model that just come out in the way it was designed. It allows for varied diploidy, so you can use all your infections, including the mixed ones. It's agnostic to the type of genetic data. We're evaluating through simulations the use of SNPs, microsatellites, and short haplotypes. We explicitly model missing nodes in the network because we know that that's going to be something that's going to happen, especially how that affects the spatial and temporal scales of between observed infections. We're directly incorporating temporal data, travel history, and potentially spatial data, although the data I'm going to show you right now are analyzed without spatial data included because we didn't want to make too many assumptions about the structure of the spatial data. And finally, the whole process gives you Bayesian inference using a MCMC. All right, so just again a few examples. So Zanzibar, this is the main island of Zanzibar. What I'm showing you here is the areas that were part of our study, just 3 districts, and they're color-coded by hot, more malaria incidence to cold, where there's very few cases. You can see transmission is very seasonal during the period of our study because of the rainy seasons. So some questions, what's going on during this dry season? There's very few cases. When you ask travel history, and as part of our study, there's a lot of travel outside Zanzibar during the dry season. Very few of the people that are presenting with malaria during the wet season traveled outside of Zanzibar. That could be just because people travel more when it's not raining, or it could have something to do with transmission. And then the second question is, what are these different clusters that are so visually apparent on the map? Are they separate? Can we do MDA or IRS or some sort of targeted intervention here, or here, or here, or here, and not tackle the other places at the same time? And would we expect to see any effect? All right, so I'm going to show you data from the entire island, but first I'm just going to zoom in on the north because it's hard to see when you zoom out too far at the spatial scales that you can present on a static map here. So this is just kind of the northern region that's indicated by this box, and I'm going to take you through the season. This is the first wet season, and I'm showing here linkages that have a posterior probability of greater than 50%, so more likely than not to have occurred. And you can see that while most of the transmission is very focal, you do have some of these stretches that are a bit further afield, connecting these apparently isolated islands. When you go from the wet to the dry season, you do see some continued connections, indicating that there is some persistence into the dry season, some transmission locally during the dry season as well, and then into the following wet season, where we then see longer scales of transmission again. So when we then take this to the entire island scale, what I'm showing here is connectivity now between these shahiyas, which are the polygons that you see on this map. On the left, I'm showing all of them. It's really hard to see much, but when you limit it to the highest transmission events, you can see distinct patterns of transmission. For instance, there's clearly evidence of a corridor of transmission between this pocket of high transmission here and this area on the northwest. And very interestingly, anecdotally, when we were doing the study, people talked a lot about migrant fishermen populations that would actually travel different parts of the coast during different seasons and could potentially be a vector for non-contiguous spread of parasites. And when you limit it to the highest connectivity, you not surprisingly get this area here where there was lots of high incidence polygons right next to each other. Trying to capture this slightly differently, looking at sources and sinks. The map on the left shows the size of the red circle, just how many cases were detected in that one area. Again, you can see the pattern replicated from before, more cases here and here. But then when you categorize based on how much malaria they appear to have exported based on these links or imported, you can see that there's not a whole lot of exportation here, but there's more importation here. So again, illustrating what some of those directional arrows showed, that there's not only connectivity between these two areas, but there seems to be a particular directionality of that. We can then aggregate, right? So instead of, if we don't care about such fine scales, but we want to look at kind of, I somewhat arbitrarily picked these 4 regions here for the purposes of this talk, to north, north central, west, and central. For those of you that know Zanzibar, north central, it's not really an administrative unit, but I just kind of made that up because it appeared to be an island. And then we use 2 different ways of comparing these. So one is just looking at pairwise genetic distance-based metrics, and this is not using FST or summary statistics for each community, but using each individual within that community and looking at all pairwise connections with individuals in other communities. We've used a couple of different distance-based metrics that allow for multiple infections. They show similar results, and you can actually bring back some of that population structure. So clearly there's a lot of transmission within, on the diagonal, within a given region, but you can see that there are certain areas of higher connectivity between central and west, and then of course between the west and north, that corridor of transmission. We then did the same thing using the network-based data, and we get similar results, but because this is now weighted by the total amount of transmission and not by the denominator of how many cases were in that area, you can see that there's a significant amount of movement in terms of from west to north and other areas. So in summary, there's evidence in Zanzibar of sustained transmission throughout all the seasons, despite few local cases in the dry season. The seasonal transmission, it seemed to be initiated from both imported as well as local cases. And most importantly, the scales of transmission really seem to indicate that highly focal interventions on this island would potentially fail. So greater than 37% of the links in the network were actually beyond the individual shihia. And most of the transmission was very local, but there were disparate regions of the island still appeared to be very well connected. All right, so I'm going to go through northern Namibia and probably skip Swaziland so there's time for questions. So this is I'm going to present part of this study. If you want to hear more data from Namibia, you should see an excellent talk by my postdoc Sophonius on Wednesday at 10:15 in the Malaria Genetics and Genomics session. So I'm going to focus on this Kavanga region. This is where we had those stickers for the health facility workers to put on with the questionnaire, and someone just drove back and forth collecting used RDTs. So when we make some networks with the health facilities color-coded from west to east along this region, you can see that you tend to see similar colors clustered together as you'd expect. When you then translate that into scales of transmission and put that against call data record data on human mobility, you see something very interesting. Just like with the human mobility data, you see most of the transmissions are occurring at relatively focal scales, but you see the tail of this seems to be a bit fatter than human mobility, indicating that transmission may be occurring on slightly broader scales. When you then put this into plots where you have coming from and going to on the rows and columns, and these are again going from west to east, you can see again if you use population level metrics like GST, there's really no signal. Everything is just kind of a mishmash. If you use the mobility data, you do see that there's more there appears to be more mobility in terms of people using different cell towers when they're right next to each other as you'd expect. But because of the catchment areas of the cell towers, you don't have quite the same granularity as we did from getting genetic data from these clinics. And whether you use the distance-based metrics or based on the networks, you can see that there are certain areas where there's broader scales of transmission occurring, and these patterns seem to be replicated despite using completely different methods, including certain squares that stand out. And these are squares where there's larger health facilities and people are probably coming from further to present to that health facility. It doesn't necessarily mean that there's more transmission in that particular area. So I'm going to skip Swaziland and just sum up here. So parasite genetics seem to be informative about parasite origin and movement at multiple spatial scales, even in sub-Saharan Africa, where traditionally there's thought to be no population structure. And this is, again, just using 26 microsatellites. There seems to be rich opportunity for if the sampling and genotyping and analysis could be coordinated across regions, so for instance, across the E8 countries. But the choice of analysis is really important. So in our data, the infection level comparisons provided much more info, even when results were aggregated to populations. And this is similar to what Amy Taylor has seen in Southeast Asia. And I think an important feature and what we hope to discuss at the end of this is that a lot of these methods are just being formulated. They need to be formalized, further developed, and synthesized across different types of data. But the good news is that the signal seems to be pretty strong, and the potential for integrating these into other surveillance metrics seems pretty high. So I'll stop there and take some questions. Thanks.
Any questions, Bryan?
Should I go?
Go ahead.
Hi, a fantastic talk and a really nice shirt, by the way.
Thank you.
So I was wondering, how much would your method benefit from going to full genome sequencing instead of using markers? And another related question is, given that you have the network for all the pairwise connectivity between infections, why don't you run some kind of community detection algorithm and see how they stratify geographically?
Those are great questions. So we haven't run it on whole genome sequencing data. It'd be great to take some simulated data or some real data to take a look. I think the model right now is geared towards relatively dense sampling, in an area of relatively low transmission. I think one of the issues for us has been because the polyclonal, over 60 to 80% of the infections, even in these low transmission areas, are polyclonal. So trying to pull out of those data something that's going to be informative is going to be a challenge. Not necessarily a challenge that's not worth pursuing, but we haven't done it yet. So we'd love to try. Right now, it assumes that the alleles are independent. So that's an assumption that could be obviously changed in the analytical method. And then your second question about detecting communities and potentially other things like temporal and spatial surfaces as part of the inferential process are things that I think would be natural extensions. I think the first thing you said we could easily do on the posterior, and that's something that we're planning to do. But I think moving forward, we'd like to actually estimate surfaces of transmission spatially and temporally as part of the inference process